Hi, I'm Miranda Jacobson, and welcome to Cellfeed. We've got a very interesting show for you tonight, but first, a quick word from our sponsors at Moz Microscopes before we begin.
For the best in microscope technology, Moz Microscopes has the highest quality, best variety, and most advanced technology in microscopes for people of all ages. Lucky for you, we are having an end-of-the-year closeout sale on some of our top-selling models. For just $999.99, you can purchase your own transmission or scanning electron microscope, where beams of electrons project onto samples, producing a 3D image of specimens. Restrictions may apply. Electron microscopes can only be used to study non-living cells and tissues. Act fast, and we will throw in 2 free compound light microscopes, whose compact frame allows us to produce images using light to magnify living samples. Though images will appear backwards and upside down, our compound light microscopes offer clear images with varying resolutions and magnifications to meet all of your scientific needs. If you place your order in the next 20 minutes, we will include a lifetime supply of slide stains to accompany your new microscope, without which the outstanding features of your samples would go undetected. Before we return to the news station, here are some images that have been submitted by Moz Microscope's customers, whose investments in our products allow them to study the incredible cells from which all living things are made.
And now we turn to Katie with the latest celebrity gossip.
Thanks, Miranda. It's been a busy week in the lives of our celebrities. First, here's a brief overview of the drama, the differences between prokaryotic, eukaryotic, plant, and animal cells. Our prokaryotic cells have been laying low for the week. They're more simple in design. Prokaryotic cells look a little like this. They have no membrane-bound organelles, no nucleus, mitochondria, or Golgi bodies. The prokaryotic cell has a systolic region on its interior where ribosomes are floating. The DNA is found in the nucleoid region of the cell, and other specialized regions within the cell carry out its normal functions. The prokaryotic cell is surrounded by a plasma membrane and have flagella that help facilitate cell mobility, and are simpler than the more dramatic eukaryotic cells, who can further be broken down into plant and animal cells, and also have many differences between them. This complexity can be supported by the endosymbiotic theory that states eukaryotic cells evolved from early prokaryotic cells, as early prokaryotic cells engulfed each other, leading to specialization and compartmentalization, giving rise to the organelles we see in eukaryotic cells, supported by the structures such as mitochondria and the chloroplasts. A typical eukaryotic cell is different from its more prokaryotic precursor, as various organelles it contains contribute to its increased metabolic efficiency and complexity. Animal cells are surrounded by a plasma membrane and contain specific organelles such as centrioles, while plant cells are surrounded by a cell wall and contain chloroplasts and a central vacuole. Well, that's it for the celebrity gossip. Now to Miranda with a special cooking segment.
Thanks, Katie! Now for our special feature, we're going to be cooking up something delicious that you absolutely need to have in your life. Today we'll be learning how to make cells. First, prepare your cell membrane made up of a phospholipid bilayer. Fill your cell membrane with chlorophyll and spread your free-floating DNA and ribosomes throughout your cell. Once you finish this step, coat the outside of the membrane in cilia and a bit of flagella onto one end of the cell, and you're all set. You've finished your prokaryotic cell. This is perfect if you're nuclei-free, vegan, or gluten-free. Next, I'll show you how to make your very own eukaryotic cell. For this, you'll need a nucleus inside of which your DNA is already pre-packaged, a rough and smooth endoplasmic reticulum, and a Golgi apparatus, some vesicles, a handful of ribosomes, the lysosome, mitochondria, and your chlorophyll filling. Our first eukaryotic cell will be an animal cell, an animal cell, an animal cell, an animal cell, an animal cell. To start your animal cell, take your cell membrane and drop in your nucleus. This organelle is one of the main differences between your prokaryotic cell and your eukaryotic cells. Next, right beside your nucleus, place your endoplasmic reticulum, rough side first. The rough endoplasmic reticulum is always laced with ribosomes, which help the cell make its necessary proteins. The smooth endoplasmic reticulum that has no ribosomes can detoxify chemicals in the cell. Next, add your Golgi apparatus, which is used to package and send off your proteins in your vesicles. Sprinkle a few vesicles throughout your cell to carry the proteins out from the cell to your other cell. Next, add your lysosome to hold any enzymes that your cell produces. Next up, add your mitochondria. The mitochondria is the powerhouse of the cell, meaning that it produces ATP energy. If you want, you can add even more mitochondria to help produce even more energy. Finally, add your chlorophyll filling. If you only want an animal cell, leave this cell as it is. Now we'll move on to the plant cell, where we'll add a cell wall to the outside of the cell membrane. This limits the semi-permeability of the cell membrane by being a thicker layer outside and also provides some sort of structure for the cell. Also, you'll need to add a vacuole, which is one of the largest organelles and is used to hold waste and materials. It also helps to maintain homeostasis. Then you'll need your chloroplasts, which allow plants to perform photosynthesis and get energy and plant food. Put these cells in the oven, wait 10 seconds, and you'll be all ready for your brand new cells. The cells are ready! I hope you enjoy your brand new cells.
Well, now I'm hungry. Thanks for that, Miranda. Before we end, we would just like to thank all of the people whose discoveries helped us know all that we know about cells now.
Thanks, Katie. Now for our special feature, we have something that you need to have in your life. Sorry, that wasn't the line.
Well, now I'm hungry. Thanks for that. I can't read.
What?
Is now the time, Miranda?
Are we going? Prokaryotic cells! Now, moving on to a plant cell, we're gonna build a wall and make the red blood cells pay for it.
It has been a busy week in the lives of our...
Thanks, Katie.